All right, welcome back. So I just want to just take a moment to just sort of touch on and review a couple concepts that were discussed or sort of brought up in those videos. And the first is sort of this idea of active versus passive immunity. So the video talked about sort of our immune system and natural infection and sort of artificial infections like a vaccination where we're taking part of a virus or a dead virus and we're injecting it to initiate an immune response. So this is both these sort of natural by actually getting the infection or these artificial types are activating our immune system, are using those different T cells that were discussed in the video, creating those memory cells so that our body will remember these different invaders, again, whether they're natural or artificial. This is sort of natural, or this is active immunity. Passive immunity is also can be natural, also can be beneficial, can also be natural or artificial, excuse me, and in this situation, we are getting antibodies passed to us from somewhere else. So these are not antibodies that we are actively creating on our own by activating our immune system. These are antibodies that we're getting passively, hence passive immunity. So examples of sort of this naturally occurring are maternal antibodies that are passed to babies through breast milk. So the mother is producing antibodies and she is passing those over to her child, again, through the breast milk. And so that baby is taking on some of those antibodies from the mother. Artificial, we can develop in a lab different what we call monoclonal antibodies that we can then either inject or we can add into the body intravenously that will act in a similar way to our own natural antibodies, and they will bind up these invaders and help our body to clear it out. So I just wanted to sort of clarify and touch on this idea of active and passive immunity. The other point that I wanted to discuss quickly is this idea of herd immunity, and we're hearing a lot about this right now with the COVID-19 pandemic. And so the idea here has to do with the percent of a population that is vaccinated. And so herd immunity is a specific threshold that we can reach. If we can get enough people vaccinated, this can minimize the spread of the disease. And so here in this diagram, you can see unvaccinated individuals are in blue, vaccinated individuals are in yellow, and we go from 0 to 25 to 50 to 75 to 90 to 95% vaccinated. And so now if we run a model where each one of these non-vaccinated individuals can pass this disease on to two other non-vaccinated individuals, and we run that model through, you can see the difference in the spread and the transmission rate in the sort of unvaccinated population versus sort of highly vaccinated populations that we see here on the bottom. So let me run this model for you, and I'm going to run through it a few times. It's going to be on repeat, but I want you to sort of focus on what's happening in this top panel, right, where we have under 50% vaccinated, and this bottom panel where we have over 50% vaccinated. Right, so you can see here as this model is running, right, and again, one individual spreads it to 2, spreads it to 4, right, and look how quickly this model is able to spread in the top, right, in this sort of less than 50% vaccinated, and look how much more slowly it's spreading down here in these highly vaccinated, right? Because if you have all of these vaccinated individuals in between, it slows the spread, it slows the progression, it slows the transmission of this virus. And in the case of COVID-19, slowing this spread, slowing this transmission, right? Every time this is transmitted, in the case of an RNA virus, there's potentials for mutations, right? So this is where the Delta variant came from. The further and faster this spreads, the more likely we are to see different mutations, different variants emerge. And so having highly vaccinated populations minimizes not only the spread, but it slows the ability of the virus to then change and mutate, allowing us to more quickly more quickly achieve a state where we can get not only this infection under control, but again, we can minimize the transmission of this to new individuals. And so now I want to talk a little bit, thinking about this idea of herd immunity and transmission from one individual to the next. As we're thinking about these different disease states, thinking about how they are transmitted is really important, right? Because not all, right, we talked about these infectious agents can be things like worms, protists, bacteria, viruses. All of these things are transmitted in different these infections is really important. Disease one individual to the next. As we're thinking about these and transmission from one about this idea. And so now I want to talk a little bit, thinking about this idea of herd immunity and transmission from one individual to the next. As we're thinking about these different disease states, thinking about how they are transmitted is really important, right? Because not all, right, we talked about these infectious agents can be things like worms, protists, bacteria, viruses. All of these things are transmitted in different ways. And so some transmission, as we know, is through closeness or contact. And so this can be things like droplet transmission. We're hearing a lot about this right now with COVID-19. COVID-19 is primarily spread through droplet transmission. And so when an individual coughs or sneezes or talks or gives a lecture or does anything where they are breathing in and out, along with the air moving in and out, small droplets of saliva are coming out that can carry virus. And if I am close to somebody or I am in their vicinity and they breathe that droplet in, they could now potentially be infected with the virus. And so, for example, droplet transmission, diphtheria, tuberculosis, pertussis, influenza, which is the flu, measles, mumps, rubella, COVID-19 are all spread through droplet transmission. Other viruses and bacteria are spread through direct oral contact. So an individual droplet isn't going to contain enough of the virus to be dangerous or transmissible. However, if you are sharing beverages, if you are, mono is a really, right, so that's sort of the kissing disease, right? If you are swapping saliva, right, again, whether that's by kissing somebody or by sharing beverages, right, drinking from the same glasses, right? So that direct oral contact, again, the virus or the bacteria that is in that saliva can be passed. So examples of direct oral contact, mono, strep, herpes simplex 1, those are all things that can be exchanged by direct oral contact. And it's not just necessarily oral contact. There are a number of diseases that contact with bodily fluids. So sexually transmitted diseases are often use other bodily fluids, or things like HIV are through contact with blood. So it doesn't have to necessarily just be direct oral contact. Contact with infected bodily fluids for a number of diseases can lead to their transmission. Other diseases can be transmitted by fomite. So fomite are just non-living solid surfaces. So if I have a virus, so the flu virus, for example, can be transmitted by doors. So I sneeze on my hands, I go to grab a door knob, I transmit some of those droplets with virus to the doorknob. Certain viruses, certain bacteria can live outside of the body for certain periods of time, right? If I open that door and then somebody else opens that door and then touches their mouth, touches their nose, right, then they are able to pick up that virus from that solid surface. So that would be transmission by fomite. And there are others that are transmitted by airborne transmissions in aerosols. Now, this gets a little tricky when we're talking about droplet versus aerosols versus airborne, right? And so you'll see here that they list this as distinct from droplet transmission. So these are all related. So these are bacteria or viruses that are transmitted through small droplets of saliva or mucus that are coming out of our mouths and nose. The difference is in the size of the droplet. So droplet transmission is the largest size, right? The largest size droplet. And so these droplets are so large that when they are put into the environment within 2 meters or 6 feet, they are not able to stay in the air. So if I sneeze out one of these large droplets, it will travel, right, no more than 2 meters before it falls out of the air and falls on to a surface, right? And that's why we have sort of social distancing recommended at 6 feet, right? Because these large droplets that we know can contain COVID-19, right, have a very hard time staying in the air beyond 6 feet. Aerosols, these are similar, but these are smaller droplets. These smaller droplets can hang in the air longer and can move farther than droplet transmission. And then airborne transmission, again, is also in droplets, but these are the smallest droplets. These airborne transmission can hang in the air much, much longer, so much so that in airborne, these teeny, teeny droplets can actually be pulled into, right, as air is circulating in the room, or if air circulation is very poor, right, these airborne transmission, these very, very small particles can stay in the air. They can be moved around through the air through different heating and air conditioning systems and things like that, right? And so these airborne transmission is, again, inside these droplets, but they're the smallest droplet. And really this has to do with how virulent a bacteria or a virus is, right? So if you have a disease that is particularly virulent, right, then it is able to hang in the air longer, and it is able to then sort of infect, right? So certain viruses, so COVID-19, for example, uses droplet transmission. It uses droplet transmission because you have to have a significant amount of the virus in order to pass it from one individual to the other, right? Whereas other diseases like measles, for example, or chickenpox, which are highly contagious, can be through airborne transmission. So even these smallest particles that contain only a small fraction or a small amount of virus, that's enough for you to catch it because they are so contagious. So again, different transmission, again, through close contact, droplet transmission, direct contact with bodily fluids, transitions by fomite, are all ways that we can transmit infectious disease. In addition to this, right, a really common path for infectious diseases is through fecal-oral transmission of diarrheal diseases. So the fecal-oral pathway is when one individual's infectious diarrheal disease becomes the next person's disease of fecal origin. So, for example, if sewage is not well controlled, right, this can lead to waterborne transmission, right? So if people's waste is going into the same water that they may be drinking, right, this can lead to this sort of fecal-oral pathway of disease, right? right? And so there are a number of examples of this. We're reading right now a lot about cholera, typhoid fever, dysentery, giardias, cryptosporidium. This is zoonotic disease, right? So this is coming from the feces of animals, and then it's moving in to our system. So these are all examples. And so this is a diagram out of your book that, again, we have human and animal feces. The primary pathway, which you can see here by the dark lines, is this feces ends up contaminating the water supply, leading to waterborne transmission. And then if you ingest this contaminated water, as we're seeing in the book, The Ghost Map, this can lead again to spreading of diseases like cholera. But human and animal feces can also get into the soil, which can then get into the water. Human and animal feces can get onto the hands, right? If you don't have proper sanitary conditions, then hand to mouth transition, again, through ingestion, we're able to bring these in to our body. So again, this is a really common mode that many diseases use, primarily, again, through the feces getting into water, and then water leading to waterborne transmission by ingesting contaminated water. There are also non-fecal organisms that can be transmitted in water and soil. So tetanus is a great example, or guinea worm disease. So again, these aren't coming from feces, but these are from contaminated soil, getting in either into the water or onto people's hands, and then going into their mouth and being ingested. And now, as we are as food in sort of modern society is becoming more and more processed, we now also have foodborne transmission as a very common path for infectious disease. And so we are regularly hearing about recalls in the grocery store. These happen on everything from meat, as I'm showing you here, to lettuce, due to E. coli poisoning, right? So E. coli is a bacteria. This is a common bacteria that is found in our gut, in our gastrointestinal tract. And if this E. coli, and it's not only our gastrointestinal tract, it's the gastrointestinal tract of most mammals, right? But if soil or water happen to become contaminated by E. coli, these can get on to meats through food processing and can become very dangerous, right? And so if we sort of insert food into this diagram, if this food is contaminated by soil that is contaminated with E. coli, right? This would be the case of like lettuce, right? If the lettuce isn't properly cleaned and processed, E. coli from the soil can stick around on the food. If that food is watered with contaminated water or exposed to contaminated water, either through sort of the growing or in the food processing phase, this can again lead to foodborne transmission. Or if this food is being processed, packaged in an unsanitary space and people's hands are dirty, this food can become contaminated, foodborne transmission, again, through ingestion. And the last one that I want to talk about, which is also really important, are vectors. So here you can see we have a fly. So we know that flies are attracted to rotting, decaying meat. They are often seen attracted to fecal material. And so if this fly is landing on fecal material and then flies over and lands on your food, this can also lead to foodborne transmission of diseases, right? And it doesn't have to just be flies. I'm using flies as an example. Flies, cockroaches, any number of things could lead to that transmission. So without sanitation, most foodborne illness is by the fecal-oral pathway, again, through either contamination through water or soil, or in unsanitary conditions through the hands of people processing the food. In industrialized countries, some foodborne illness is of human fecal origin, so shellfish contaminated by raw sewage, inadequate hand washing in food preparation. We are very familiar with this a little bit in Chicago, where beaches are closed because of E. coli contamination, right? So this is, again, some sewage has gotten into Lake Michigan, and E. coli levels are such that people can't swim because they could contract E. coli. But again, most foodborne illness is from other sources, right? Animal fecal pathogens, if animals are improperly slaughtered in the preparation of meat, and some of the gut contents contaminate the meat, right? That can lead to foodborne illness. Pathogens in the soil, on food, right? Again, if the lettuce isn't properly washed or prepared, this could lead to pathogens. Human skin, again, through the preparation, if appropriate sanitation isn't observed, right? Human skin can contaminate these food sources, or mechanical vectors such as flies and cockroaches can carry bacteria in if, again, this is not highly regulated. And so there are a number of basic levers for food safety, right? So we regulate food to help minimize this transmission of infectious disease. So for example, time and temperature are really important, right? There's an acronym: keep it hot, or keep it cold, or don't keep it. So if you're cooking your meat, right, you read the back of any meat, right? It talks about how it's not safe to eat raw or unprocessed meat. You want to cook it. If you cook the meat appropriately to the appropriate temperatures, you're going to kill any bacteria that could be contaminating it. Or when you bring meat home, you put it in the refrigerator, right? This is going to minimize, again, the ability of this bacteria and things to grow until you can appropriately cook it to consume it. So again, time and temperature are really important. And if you have something that you think hasn't been cooked well enough or hasn't been kept cold enough, best advice is to not keep it. The temperature danger zone is between 40 degrees and 140. So you want to either keep it under 40 degrees or over 140. And again, for different meats, this ranges. 140 for my personal taste is a little bit low. For example, if I'm cooking beef, I like to cook that to at least 165 degrees, if not 170 degrees. And again, making sure you're keeping your refrigerator cold enough so that it's below 40 degrees is important for maintaining appropriate food safety. And then we also want to, again, the timing is really important, right? Bacteria have a very characteristic growth in their population. And if we can minimize that exponential growth and hold them in their sort of lag phase, then that's, again, going to keep those bacteria levels low. And then it's going to allow for us to be able to then use temperature, right, in the case of either keeping it hot or keeping it cold, to minimize the spread of those bacteria. So again, a really important sort of component now in modern society is in food preparation, where we are getting a lot of processed, prepackaged food, right? Ensuring that processing is regulated and done in a safe way is really important to minimize foodborne transmission of infectious diseases. And so here is, again, another table from your textbook. Estimating the overall incidence and case fatality ratio for the 4 most common foodborne illnesses in the United States. So I brought up E. coli already. Listeria is another one. Salmonella we're very familiar with. This is salmonella is found in the gut and gastrointestinal tract of many reptiles and birds. So salmonella can be from eating raw eggs, for example. And Campylobacter is another one. And again, showing the incidence per 100,000 in the population. And again, the fatality rates, again, are lower. But again, these can be really important if they are not appropriately regulated. So we also talked a little bit when we're talking about food, these vectors, right? So how flies might sort of interact with fecal material and then come land on your food and transfer infectious disease into the food, right? It's not just with food where we see this vector-borne transmission. There are a number of biological vectors, host species that transmit diseases onto other host species. Many of these vectors are arthropods, they're insects or arachnids, but mammals can be vectors too. So for example, rats and mice can carry infectious disease. And so it's important that we minimize their levels, because if their levels rise too high, they can be transferring some of these diseases into human populations. But speaking of the arthropods, the insects, two really common examples are mosquitoes. So for example, dengue fever or West Nile virus is transmitted from one individual to another through the bite of a mosquito. Or black-legged ticks, for example, are known to carry Lyme disease and transmit this through their bite to humans. So again, these are the ticks are picking up Lyme disease from usually mice as the reservoir. Lyme disease doesn't affect mice in the same way it affects humans. So they bite a mouse, they get this bacteria into their system, and then when they bite a human later on in life, they can pass on this Lyme disease. The same with the mosquitoes. West Nile virus, I believe, is that when they bit a bird and then they bit humans, they can move West Nile virus from bird reservoirs into humans. So again, vector-borne transmission is really important. And the reason that we're going through this is, again, remember, thinking about these different potential sort of environmental contaminants, right? Understanding the fate in transport, how these are moving through the environment, is really important for us to create models and to understand how to address these different environmental health issues. And so here I'm showing you sort of a very, very complex lifestyle of Rift Valley fever virus that has a number of different reservoirs, right? So it's being held sort of in the sylvatic cycle in sort of mosquitoes, transferring it around to wildlife, right? Those same mosquitoes can then pass this into domestic animals, and then those mosquitoes can pass it from domestic animals in sort of peri-urban mosquitoes into human populations via zoonosis, right? But you can see that there's a complex set of arrows here that are showing how this virus can move through different populations in an ecosystem and eventually get into humans. And so understanding where the reservoirs are, what animals are holding this disease but not getting sick, what are the organisms, what are the mechanisms that it's using to transfer? These are all things that we could potentially regulate or attempt to address to minimize the spread of these diseases. So this then leads to, right, sort of understanding these models, understanding the fate in transport, then leads to the sort of evolution of strategies for managing the transmission of disease, right? So segregation of sick or exposed individuals. So isolation, quarantine, self-quarantine. These are things that we're very familiar with now. So this is one strategy. If you have sick individuals, keep them away from the population, minimize their ability to spread this to someone else. So we can either isolate them away from everyone, or we can quarantine them in a space that keeps them away from the general population. Sanitation is also really important. If we're talking about fecal-oral transmission, sort of appropriate sanitation, not only appropriate sewer systems, but also how how those waste management systems are processing that waste, what's happening to the waste, how are they sterilizing the waste. Sanitation can be hand washing, right? We know right now hand washing, as we're sort of moving into the fall, as we're moving into flu season, as we're dealing with the coronavirus, right? Washing your hands is very, very important. So not only sort of personal sanitation, but also sanitation in our public spaces, right? Keeping things clean in our food preparation spaces. These are all important strategies that help minimize disease spread and the transmission of infectious disease, right? We also now have vaccinations, again, something we're very familiar with to prevent illness. If we can vaccinate, if we can move towards herd immunity, this is going to slow the transmission of infectious disease. We can also use antibiotics to treat illness, right? Antibiotics can be given for certain bacterial infections that will minimize them, potentially destroy them. However, there are some risks with antibiotics, right? Certain populations of pathogens can become resistant over time. So we're seeing strains of what we call MRSA, right? These are a staphylococcus, right? This is a staph infection. This is a bacterial infection, but these are methicillin resistant. This is like the one antibiotic that works very, very well against staphylococcus, but many staphylococcus infections are becoming resistant to this one antibiotic that we have that is effective. And so, again, we can use antibiotics, but we need to be careful that we're not creating worse strains of these bacteria. Pesticides are another strategy that has been used to control vectors. So if we can control the mosquito populations, if we can keep down the populations of cockroaches, if we can control the levels of rats and mice in an environment, we can control the vectors, and again, we can help to manage the transmission of these different diseases. Right, again, managing, I already sort of covered this, managing vector-borne transmission, preventing human contact with the vectors. So through clothing, screens, nets, insect repellents can prevent us from coming in contact with those ticks, those mosquitoes, right? If you are in an area where malaria is really prevalent, you may sleep under a mosquito net to keep mosquitoes away from you at night. We can also reduce the vector populations through pesticides, modifications to the environment, making it less favorable, or in the case now, we are seeing some release of genetically modified sterile male insects to reduce reproduction. So introducing these sterile males acts a little bit like the herd immunity, right? So if we have these males that are competing for mates, but then are sterile and cannot actually create mates, that's going to end up slowing the spread by slowing the reproductive capacity of these particular populations. So again, different strategies for managing vector-borne transmission. And what I really want you to take away from this is that transmission is a complex web. And so I'll sort of just put back up here the Rift Valley fever virus as a reminder. It's not always clear. Distinctions among modes of transition are often blurred. And we could think about that with COVID-19. Yes, it's primarily through droplet transmission, right? But that doesn't mean that it couldn't land on a surface. It's less likely, but it could, right? Different diseases are going to show different modes of transition. Some of them are going to have multiple modes of transition. And so we need to think about this as we're thinking about transition, again, to put in the appropriate regulations. Transmission is also not always accidental. We are seeing different pathogens used as weapons by the military, by terrorists, as biological weapons, where they are releasing strains of infectious disease on purpose. We're also seeing emerging or re-emerging infectious diseases. So things like Ebola, hemorrhagic fever, dengue fever, hantavirus, the sort of E. coli that I was talking about, drug-resistant malaria. So understanding these complex webs, so not all E. coli are created equal, not all influenza is the same. And so thinking again about these complex webs is going to be very important, because what we want to think about is, again, now that we understand the fate and transport, right, what kind of policies, what kind of regulations, what kind of preventative measures can we put in place to minimize these spreads, right? And so thinking again about understanding the cycle and then being able to put in regulations. And so the U.S. regulatory framework for managing infectious disease is through vaccination. So to attend school, you have to have certain vaccinations. The CDC develops guidelines about which vaccinations should be required, and then states implement that, again, by requiring them for you to come to school and things like that. So again, vaccinations are regulated by the CDC or recommended by the CDC, and then different states implement that. Isolation and quarantine, again, this is nationally done by the CDC and individual states within their own borders. We have seen a lot of this with the COVID-19 pandemic. So nationally, the CDC may close borders. Within states, we could see lockdowns like we saw in Illinois with sort of self-quarantine orders. CDC is also surveilling listed infectious diseases, right? So they're collecting data state by state. So if a disease shows up in a hospital in Chicago, they communicate it to the CDC, and the CDC is monitoring these levels to potentially, if there is an issue or there is a problem or this doesn't appear to be an isolated incident, they know this and they understand it, and they're helping not only to watch within our own borders, but also worldwide and understand where infectious diseases are, where they're coming from, and helping to keep us safe. In addition to what's happening sort of with the CDC, we also have very strong regulation of the food supply. So the USDA and other organizations regulate our food supply. There's regulations on how we treat sewage and drinking water. All of these are important regulations that, again, are targeting the management and the prevention of the spread of infectious disease. And so again, when we're thinking about these complex webs, then we want to be thinking about these mitigation strategies. The World Health Organization is also really important, and they are not only reacting, right, but they are working on anticipating emerging infectious disease epidemics. And so this is sort of an infographic on anticipating emerging infectious diseases and the complexity and sort of the multifaceted nature of that. But what I want to point you to sort of two systems, right? The first is sort of what we've been talking about, right? So sort of looking at the context, right? What's the severity? What's the frequency of occurrences of particular pathogens, right? How do we take that information? How do we anticipate and define the drivers? How is this being transmitted? How is this being moved around? What is the fate in transport? And then how do we use that information to move us in the right direction, right? How do we develop collaborations in emerging technologies and the community engagement that we need to prevent the spread of these particular diseases? So I feel like this is really sort of important and really sort of highlights what we've been talking about here. I think the other is, again, thinking about how can we use an organization to be better prepared, right? Sort of what are levels, how can we leverage expertise, right? How do we implement responses? What are the different information systems that are being used, right? And we are learning from different pandemics. We are learning right now from COVID how to better respond to emerging infectious diseases. This one here is highlighting Ebola, right? We learned a lot from the spread of Ebola and how we as a society responded, right? And the sort of old methods aren't, some of them are good, but they are not always the most effective. And so as you all are moving forward and moving into public health, right, you need to be engaging all of your stakeholders. You need to be thinking ahead, thinking outside the box, leveraging expertise to be able to think about these sort of complex webs of transmission and how can we step in and create preventative measures to prevent them from, prevent the further spread of these infectious diseases. And so with that, I want to just change gears to sort of the next part, right? So we just talked about infectious disease, and now I want to have you watch 4 videos, I believe, that are going to discuss sort of the next 3 areas in this chapter. Poisons in nature, so we talked about infectious diseases. Now we want to talk a little bit about different poisons that are naturally occurring. And again, I want you to think about those in some of the same ways, right? There are poisons, there are toxins out there. Thinking about how we manage where these are, what they're doing, how we're using them, right? This is going to be important. And then again, thinking about not letting sort of these naturally dangerous agents spread. So thinking about poisons in nature, talking a little bit about allergies and asthma, right? This can be related to environmental health. So allergies and asthma can be triggered by things like mold and different spores and pollen and things in the environment. Again, how do we think about that? How do we think about the sort of the transport of these different allergens and asthma? And then the last one that I want to talk about for this section is sort of natural disasters. And thinking about natural disasters, again, these are natural phenomenon, but then these natural disasters often result then in very, very powerful environmental health issues. And so I want to just talk a little bit about talk a little bit about natural disasters, and I want you to think for a minute about how that might lead to different environmental health outcomes. So now if you want to flip over to the 4 next videos in D2L, and you can watch those, and then we'll come back and talk a little bit about radiation. All right, welcome back. So I just want to take a All right, the last thing that we want to talk about this week is naturally occurring sources of radiation as environmental hazards. And so radiation occurs in a lot of different forms, and these forms are called electromagnetic radiation. And so this is energy that is traveling in the form of a wave, right? So you imagine that wave, and waves can take on different shapes. The shorter the wavelength, the shorter and faster the wavelength, the higher the energy, and the longer and slower the wavelength, the lower the energy. And so the electromagnetic radiation that we're going to be the most concerned with are those of the shortest wavelengths, things like gamma radiation, which are going to be very, very short wavelength, very, very high energy, and can be very damaging to living tissue. But remember that the electromagnetic spectrum is very broad. There are a large number of different wavelengths that are occurring. And so these range from very, very long, low energy wavelengths like radio waves, which we use for the radio. You're using this technology for your wireless internet, your cell phone. These are the radio waves. Then we get a little shorter. We move into microwaves. This is what you're using in your kitchen to cook your food. We get a little shorter. Infrared. This is heat that's sort of being released off of you. If you're thinking about an infrared camera, infrared cameras pick up these sort of low energy wavelengths here. We then move into this very small window in here, which is visible light, right? Your Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. This is going to be the visible light spectrum that we can see is this little teeny narrow window. Then this moves into ultraviolet. This is going to be sort of, now we're moving into more stronger wavelength, shorter, more powerful energy. So ultraviolet radiation is what gives you a sunburn if you spend too much time out in the sun. We then move into X-rays, right? These are going to be the X-rays that they use in the airport to scan your luggage, or they're using in medical technology to look at your bones, right? So these are high enough energy particles that they are able to pass straight through your tissue, your soft tissue, muscles, and skin, but they don't pass through bone. And so they allow us to look at the bones through the tissues. So those are X-rays. And then we get to the shortest wavelengths, the most powerful electromagnetic radiation, which is going to be our gamma rays, which comes in different alpha and beta particles that we're going to talk about here in a minute. So again, this all has to do with the wavelength. The shorter the wavelength, the higher the energy. The longer the wavelength, the lower the energy. So it's really these short wave, very high energy radiation that we are most interested in and can be the most dangerous. So again, as I mentioned, this radiation is energy traveling in waves. In physics, this also can get complicated. It can also act like a particle. So you could think about these waves as they're moving, right? These have energy. They have the ability to do damage. And so the energy can be thought of as a particle or a wave. For our purposes, it doesn't really matter which way. I would think about them as waves, right? But you could also think about them as particles moving in a wave form. And the most powerful forms of radioactive energy are coming from radioactive decay. So when we have really large atoms, sometimes the nuclei of those are so large that they become unstable. And so these are often called radioactive elements, right? And so these unstable isotopes try to stabilize themselves by ejecting part of their nucleus. Their nucleus can't hold together, and as it falls apart, it ends up releasing either alpha or beta particles. That's these really high energy gamma radiation. And so the alpha particles are 2 protons and 2 neutrons that fall out of the nucleus and are ejected, and they come out at very, very high speed, very high energy, giving us, again, a form of gamma radiation. And the other is a beta particle, which is an electron that is being ejected from this atom. But that electron is coming not from sort of the electrons that are orbiting around that nucleus. This electron is coming from the decay of a neutron into a proton, or a proton into a neutron. So again, as that nucleus of that radioactive particle is decaying, some of these neutrons become unstable and convert to protons. And when they do that, they release a beta particle or an electron. Or a proton becomes unstable and converts to a neutron, and they release one of these electrons as part of that, which is the beta particle. And so as we change the number of protons, right, one element decays into a different element. And this decay occurs in a characteristic series. It doesn't just randomly jump around, right? As it shrinks, as that nucleus gets smaller and smaller and smaller, these different radioactive isotopes work their way and decay in a very characteristic series. And each element in this decay has its own half-life, right? The duration that it takes for it to transition from that particular element to the next one down. And so when we think about radioactive isotopes, uranium is one that obviously we use for nuclear energy. We use this for atomic weapons, right? And so this uranium is a great example to think of. And so here is the radioactive decay of uranium-238. So we can see here that as uranium-238 releases an alpha particle, it's going to go to thorium. As it releases two beta particles, it's going to go to now uranium-234. Then it's going to go down to, as it releases more alpha particles, to radium, to radon, down to lead, to bismuth. And then eventually, if we wait long enough, this uranium-238 will eventually work its way down to lead-206, which is the stable molecule. And so I mentioned that each one of these transitions has a half-life, right? Has a time that it takes for this to happen. And you'll notice for uranium-238 to release that alpha particle, right? Uranium-238 will release alpha particles for 4.47 billion years, right? And then when it starts to degrade, then thorium is 24 days. This then can decay for minutes, right? Then we're back to uranium-234, which is thousands of years, right, is the half-life. Down to radium, again, thousands of years. Then we're back to days, minutes in this very, very quick transition. Down, again, lead, 22 years from lead-210, right? So the key here is you can see that these molecules are producing these alpha particles, they're releasing this gamma radiation, and they can release it for very, very, very long periods of time. So if we end up having an accident where these are released into the environment, or we have soils that end up being contaminated or waterways that end up being contaminated, it takes a very, very long time for that radioactive energy to be released and for these to degrade down into some stable form. So these particular radioactive isotopes are very, very powerful environmental hazards that can have long-term environmental repercussions, right? And so really, I keep talking about these short wavelength and how powerful and how damaging this can be. And this really gets into the last piece that I want to talk about today, which is ionizing versus non-ionizing radiation. And so the big distinction here is, again, on how powerful these waves are. And so ionizing radiation is radiation that is strong enough that when it strikes matter or strikes another atom, that it has enough energy to knock an electron out of orbit, right? So imagine this wave that's so powerful when it hits another atom, it's strong enough to knock an electron out of its orbit, creating an ion. And in the case of human cells, this ionization is very, very dangerous and can lead to cell damage, specifically with our DNA. So if we have a wavelength that is so powerful that it can come in and it makes contact with an atom in our DNA, it can lead to breaks. It can lead to double-strand breaks. It can actually, if it's powerful enough, it can slice our DNA into two pieces. And in the repair of this damage, whether it's just making a little nick in the DNA, it's creating an ion, or it's leading to these double-strand breaks, this then leads to mutations, which can cause downstream damage or decay if the cell is able to survive at all. If a cell encounters high enough levels of ionizing radiation, the damage is so bad that the cells cannot survive. And so alpha, beta, and gamma radiation are all considered ionizing. So if we look back at our electromagnetic spectrum, here's our radio waves, these longest wavelengths, microwaves, infrared radiation, visible light is all non-ionizing. So these are all safe forms of electromagnetic radiation that are not strong enough to displace electrons, and so these are not going to lead to any kind of cell damage. Now we know that ultraviolet radiation, if we the very lowest forms, right? So you can kind of see that this line is a little bit, right, sort of as we transition from the very, very edge of that visible light into ultraviolet, this is non-ionizing, but by the time we reach the sort of extreme end of ultraviolet radiation, we now have flipped to ionizing. And this is where if you end up getting a really bad sunburn, right, that burn is because that the ultraviolet radiation has damaged your cells. And as that sunburn heals, right, a way that our body heals that is your skin ends up peeling, right? So some of those cells take on so much damage that they're unable to survive, and then our body works to sort of slough those off. But we also know that if you spend too much time out in the sun, you don't have your sunscreen, too much ultraviolet radiation can lead to DNA damage, which can lead to skin cancer, right? X-rays, gamma radiation are also going to be ionizing, right? This is why we try to minimize the number of X-rays that you get, right? Your dentist only X-rays you now every 2 years as opposed to every 6 months when you came in, because we know that these X-rays, if you're getting too many, right? So again, small amounts is okay. Your body can probably recover from this, but if you're getting X-rays every single day, right, this could end up leading to damage to your cells. And then the gamma radiation, like I was talking about, those alpha and beta particles are also going to be ionizing. So I have one last video that I'd love for you all to watch on radiation and how radiation is created. And this will end our lecture material for this week. And so I look forward to having you all watch the videos again next week.